Talking about conservation, first let's have a look at the big framework worldwide in terms of the global strategy for plant conservation. We cannot survive without plants, and plant conservation has been a global topic. And the Global Strategy for Plant Conservation 2011 to 2020 is the framework that I have been referring to since the latest version. The new version was only formally published last year in October. While looking at this framework, the two of the five objectives are the main ones that are leading the research structure of my PhD journey. So both of the objective 1 and 2 focus on the plant diversity to be well understood, documented, and also the plant diversity urgently and effectively conserved. And with these targets in mind, and also referring to the literature with the framework of the integrated plant conservation, I have adapted the whole structure into this workflow. And the conservation I am focusing on is the ex situ conservation, which means conserving the plants out of their natural habitat. And there are several main aspects, including germplasm conservation, understanding living plants and propagating them in the botanical gardens, and then doing research on taxonomy, biology, and also ecology of the species that we conserve, as well as educating the public and also collaborating with the government also, to raise awareness on plant conservation. And in the world of rhododendron conservation, I have been closely collaborating with the BGCI, which is Botanical Gardens Conservation International, which is the international organization that looks at the conservation for all the threatened species, working with all the botanical gardens worldwide. And then the Global Conservation Consortium for rhododendron is the one focusing on the rhododendron species. And these two have made great contribution to the data collection and analysis during my PhD study. So I mentioned that I am looking at the rhododendron species conserved out of their natural habitat, which is ex situ conservation. First, let's have a look at the world view of all the rhododendrons in cultivation worldwide. Here is a map that I collected from the GBIF website, which records all the rhododendron collections, including also virtual specimens, across all the areas worldwide. And as you can see, rhododendron is cultivated all over the world in all these continents, except for Antarctica. And the majority of the plants are cultivated in the Northern Hemisphere. And then looking back to the origins of these rhododendron plants, here is a map showing the natural distribution of rhododendron species. So here we find that the rhododendron species grow naturally mostly in the northern hemisphere again, and with the greatest species diversity from the southern Himalaya to southwestern China in the red area here on the map. While as in South America, I'm just trying to show the pointer. So while as in South America and Africa, there's no native species. So during all this time, rhododendron plants have traveled from their homeland to some other land. So for example, from China to New Zealand in this corner down of the map. And it is exactly like myself migrating from China to New Zealand. So here I am showing two pictures of the rhododendron flowers. The first one on the top is one wildflower that I captured in my hometown last year. And I was very surprised to see the rhododendron flowers in their natural status. But then in New Zealand, we are also enjoying the glamorous flowers in many of the gardens, and the visitors are very happy and love to enjoy the glamour of these rhododendron flowers. However, rhododendron does not only have the horticultural function. It is not just because it's beautiful that we love them. It also has many other functions. For example, rhododendron is a cultural symbol as the national flower of Nepal. And also people have been producing mad honey from the nectar of the Rhododendron ponticum species. Also, people have been producing some of the handcrafts from the timber of the rhododendron plant. Economically, rhododendron plants form a very crucial part of the alpine ecology, sorry. So, as you can see in this graph that I referred from the literature, so a diversity of the rhododendron species grow in different elevations levels of the alpine habitats, and they also contribute to the insect pollination. So the rhododendron plants together, they have horticultural function, economic function, and also very importantly, ecological function. However, with many of us being aware of the destruction of the natural habitats of rhododendron species, and also there's a lot of knowledge gaps that we learned from the previous conservation assessment for the rhododendron species. There's a lot of work for us to do. In 2011, the Red List of rhododendron species, rhododendrons was published, which is quite a while ago. This Red List reported that over a quarter of the more than 1,000 rhododendron species is threatened, and another 23% of the rhododendron species lack data on their conservation status assessment. So a main problem of the rhododendron conservation assessment is that many of them have the problematic species delimitation, which hinders the conservation assessment, and that's one of the main reasons that's why the conservation assessment is going very slow. In 2011, my PhD supervisor, Mariam Mackay, who was not my supervisor yet by then, led a team updating the global ex situ collections for rhododendron species. So this report looked at the overview of the global ex situ collections across the various botanical gardens worldwide. And it highlighted some of the conservation issues and gaps in the by then botanical garden practice. However, this report did not look at the particular species and how they are conserved in the gardens. So following this previous work, I focused on the species level analysis and used the subsection Maddenia as a case study among all these species. Briefly introduction about the subsection Maddenia is, it is in subgenus Rhododendron, section Rhododendron. All these Maddenia species are lepidots, which means they have scaly leaves and flowers, and most of them grow in the temperate forest from the Himalayan mountains. And very importantly, these Maddenia species that present a very high morphological variation, which causes a lot of confusion in our taxonomic identification. And this is also the main reason that I have been working on this group of species. And a highlight is that the Maddenia flowers are very fragrant. This group is reported to be the group having the greatest number of the fragrant species. And benefiting from the sufficient number of the Maddenia species conserved in New Zealand, I have been collecting samples from many of the gardens in New Zealand, and also collaborating with Royal Botanic Garden, Edinburgh, as well as Rhododendron Species Botanical Garden in Seattle, to form this international collaboration to conduct my research. And here is an overview of the research structure. It might be looking a little bit complicated, but basically, under the big umbrella of plant conservation and focusing on rhododendron ex situ conservation, I looked at the wild diversity conserved in global collections to find out some of the conservation gaps in our current ex situ conservation. And then I focused on the taxonomy using modern technologies to analyze the molecular phylogeny of the Maddenia species, so that it helps us to understand the species relationships of this group. And then with these two studies, it helps us to prioritize the species in the conservation management. And then with the species prioritized, I selected a few species as a case study for horticultural practice in our garden practice. So I did controlled pollination to understand the breeding strategy for each individual species. And then together we formed a plan for how to conserve these species in terms of their germplasm conservation. So this is the whole framework in terms of the workflow that I run for my PhD research, and also a combination of the theoretical research as well as horticultural practice in the garden conservation. And in this framework, I mainly focused on answering these 4 questions. Question 1 is, what are the ex situ conservation gaps for subsection Maddenia? Question 2, what ploidy variation exists in rhododendron, and particularly subsection Maddenia? The answer to the question 2 is prep for answering the question 3, because they are together the genetic work, and I will explain a bit about why I had to look at the ploidy variation later. So the question 3 was to answer, how can molecular phylogeny help species delimitation for conservation? And lastly, can controlled pollination be used to maintain biodiversity of species in living collections? So for answering question 1 about the ex situ conservation gaps for subsection Maddenia, first, to study this group, we would have to figure out how many species that we are studying and we are analyzing. So I had to first update a checklist for subsection Maddenia and generating a taxon profile for all the 65 taxa. And then I created these maps to look at each individual taxon. So on the left, as you can see, it shows the taxon richness in the countries of origin for all these species. This is an overview of the whole group, but then in my publication, there is a profile for each of these taxon. And as you can see, the Maddenia species grow naturally in 9 countries, with China having more than half of the native species. And then on the right is a global view of all the ex situ collections of subsection Maddenia. And as you can see, North America, Europe, and then Southeast Asia, so this is the area of China, and then Australia and New Zealand have many of the ex situ collections of subsection Maddenia. So I looked at the ex situ collections at the taxon level and found out this list of the countries have the living collections of subsection Maddenia. Among them, New Zealand, Australia, US, and UK have the greatest number of the collections. And the largest number of taxa are conserved in these 7 botanical gardens. a highlight is that New Zealand has two of the main botanical gardens conserving a big portion or proportion of the Maddenia collections. And another highlight here that I'd like to address is that China is the only country of origin having living collections of subsection Maddenia, while as the other eight are have only reported conserving the species in situ, so on site. And then further, I looked at the origin of the wild source collections at an accession level. So here is a map of where subsection Maddenia come from in the origin in the orange area. And these dots show where the ex situ collections from worldwide actually came from in the wild distribution. So by doing this, I could use the geographic representation of these ex situ collections as a proxy for their genetic representation. So for example, subsection Maddenia grow in this orange area, and our ex situ collections came from these dotted localities. And as you can see, this overview shows that subsection Maddenia has decent genetic representation. And our results tell us that 55 of the 65 taxa are in cultivation, which is a good theme, and it meets the global strategy for plant conservation target 8. However, if we look deeper on each individual species, then we found 16 of the 65 taxa have no wild accessions. And even if we knew that some of them were documented or noted as from the wild, but they lacked provenance data. So there was 35 of the wild accessions that I could not map them, simply because that during all this documentation process, we have lost the information in terms of their provenance data. So this leaves a big gap for us to think about that for many of the species, even if we have collections in our gardens, we have lost the information, or we have no idea whether they are wild or not, or we have lost the information of their wild source. So the big gap for us to put into action is that we probably need to collect more wild plants or material and then back them up in our botanical gardens. So this is part 1 of my studies. And then part 2 is to analyze the ploidy variation in rhododendron species, particularly in subsection Maddenia. The reason that I had to analyze the ploidy levels of subsection Maddenia species is because before we look at and analyze the sequences for these species, if we ever have the polyploids in our samples, then it's going to cause some troubles, because the polyploids with their multiple chromosome sets, they will cause challenges and difficulties in our pipeline analyzing the sequences. So an ideal workflow is to figure out the diploid levels for the samples and then separate the samples in the pipeline for the molecular phylogenetic study. And by reading the literature, I had found that in the whole genus of rhododendron, some of the species were reported to be polyploids. And people had reported that there are some polyploids in the subsection Maddenia. So I had to first analyze the ploidy levels of my samples. By using the combination of modern technology, flow cytometry, which is very efficient and less time consuming, and then validating the data with chromosome counting, which is much more time consuming and very demanding in terms of the timing of collecting the samples. Good news is that I found out all the tested Maddenia species are actually diploid, except for the type species Rhododendron Maddenia. This species is a species complex. It is having a very close relationship with some other species, and itself is a combination of many synonymous species. So in my analysis, I did separate these species and take the samples out and analyze in a little bit different and separate work stream. One general message is that we have all these diploid taxa from the Maddenia species, except for Rhododendron Maddenia. And I did have also a review of all the ploidy levels reported in rhododendron. So there's a whole lot of information from this analysis and literature review, and they have been published in my paper, which you could find in the reference. And with the ploidy level information, then further, my task was to answer the question, how can molecular phylogeny help species delimitation for conservation? So I collect leaf samples from different gardens in New Zealand and then from Edinburgh, from Seattle, to have a better coverage of all these species in subsection Maddenia. And together, I sampled 40 taxa out of the 65, including some of the polyploid samples from the Rhododendron Maddenia species, and I used target capture sequencing. And here is a graph showing the results of the reconstructed phylogenetic tree of subsection Maddenia. The words here in this tree are very small, but that's all good. So this tree here is just to give you an overview of the structure and the different branches of the whole group. And on the right, I have labeled some of the big blocks in the whole subsection Maddenia. And this is from the traditional classification that was done by Davidian in 1982, who only used the morphological classification. So by comparing my phylogenetic results with the morphological classification, the main findings from my study is that most of the recognized subsection Maddenia taxa are monophyletic, which means the multiple samples of the same species, they landed in the neighbor branches in the tree, which is a good thing. Means, okay, this species is very simple and it is recognized and it is not getting a lot of confusion to us to identify it and also to focus on it and decide to focus on it and decide the conservation strategy on this species. However, we did find a few taxa paraphyletic, which means the multiple samples of the same species might be landing in scattered branches across the tree. So that will cause a few troubles to us in the conservation decision making. We will not be very sure about how to treat these species because they are having a close relationship to other neighbor species, and whether we should split the different branches of the species or put them together and consider as one species. And also, further up, I did find that subsection Maddenia is likely to be not monophyletic. So if you look at this tree from the root to the branches, especially at the boundary area, the species from subsection Maddenia are embedded with a few other species from the close subsections. So that's why I'm saying subsection Maddenia is likely not monophyletic. So that brings us another big question on whether subsection Maddenia should be considered as a single group itself. Now we have layers of question in terms of this group and in terms of the species within this group. Because from all the time we have been calling subsection Maddenia, but now we are actually having a little bit doubt on whether it should be subsection Maddenia or not. So from this study, in my manuscript and my thesis, I have provided some suggestions on how to treat this group and the species that we have found out that are paraphyletic, and further formed some conservation suggestions on how to treat them, and then make decisions on how to assess the conservation status for these species. So this fills our knowledge gap on how to make decisions on prioritizing the Maddenia species. And then with a selection of Maddenia species, as I mentioned, further, I conducted controlled pollination and to understand whether it can be used to maintain wild diversity of species in our living collections. So strategic thinking from this practical work is that when we conduct controlled pollination and to decide whether we can use selfing or outcrossing within the species to generate offspring for the species or not. So, for example, if we can use selfing or outcrossing to exchange the genetic material within the same species, then it's a very good thing. It means that across different botanic gardens, we can pollinate the different plants of the same species and then maintain the biodiversity level that is conserved in our botanic gardens. However, if this species is not able to produce seeds, or even if they can, but the seeds are not viable, then it means the strategy of exchanging the pollens and the plant material of the same species is not workable in our botanic gardens. So we will have to collect more wild plants or material from their natural habitat so that we can back up the wild diversity of these species in our gardens. So with the selected 5 taxa that I studied, here is a picture showing the process that how I conducted the controlled pollination using the vase to collect and dry the pollens, then dip the stigmas into the pollen to get them pollinated, and then tape the stigmas, and then waited for some time to see if they can set the seeds. So with the seed pods that I collected, then further I looked at the seeds under microscope and then did the seed germination test to see if they actually can produce seedlings or not. So using this seed germination test as a proxy for the seed viability. So with the 5 taxa that I investigated, the high-level finding is that each of them actually requires a specific study and reproductive strategy, because there was one didn't set seeds, there was also one produced seeds, but they were quite empty, so no seedlings coming out from the germination. For example, this one could produce seedlings actually. So a recommendation for our horticultural practice in the garden conservation is that if we decide that this species is to be prioritized for our ex situ conservation, and we are trying to use controlled pollination for horticultural practice and conserve the wild diversity of this species, then we would have to study this species particularly and then form a reproductive strategy. And since my study was the first time using X-ray image to look at the rhododendron seeds, and I found it was not very optimal yet, and it was not very suitable to look at rhododendron seeds as a proxy for viability, mainly because the rhododendron seeds are quite small, if you have seen that in the gardens. But I do believe, with my own experience, I do believe that there's a space of optimization to better improve the resolution, and possibly we could use this technology to look at the seed fullness of the rhododendron species. So above are all the 4 studies that I did in my PhD, and so the research covered on different aspects. in terms of the wild populations, and then taxonomy and the breeding strategies. So this is a main block towards the integrated conservation for rhododendron. And my research also closely involved in the living plants in the botanical gardens, and there was quite a lot of identification work, and also horticulture in terms of using controlled pollination to conserve the genetic diversity in the botanical gardens. And it also touched on the germplasm to back up the seeds in our seed bank and maintain their viability level. And another main block is education, which is to give trainings to the local people in the natural distribution, and also raise public awareness worldwide, collaborating with government and schools. And then as a researcher, we publish our research findings and give presentations to the public. So following my PhD, and I have been trying to get full coverage of all these aspects. So in the two main projects that I have been involved in this is the first one ongoing, which is red listing rhododendron subsection Maddenia. So this is a work that I have been working with the IUCN Red List group to gather information on this list of different data for each of the species. And this is based on the conservation gap analysis and the checklist that I generated for subsection Maddenia. So I am making the good use of my own studies. And then this work, conducting this work, I have been working closely with Alan Elliott from from Royal Botanic Garden, Edinburgh. As many of us or you have known, the conservation assessment for rhododendron species has been very slow, and it hasn't been updated since the publication of the Red List in 2011. And main difficulties include the difficult access to the wild populations in the wild, and the other is the taxonomic confusion for many of the species. So while this project is ongoing, we would love to hear from you if you would like to contribute, or if you are interested in helping us to gather some data for any of the rhododendron species, so that we can work together on the Red List assessment. And the second project or activity that I have been doing is the National Conservation Project in New Zealand. So for the rest of my presentation, I will talk about what's happening in New Zealand briefly. So here, looking at the two cropped maps that I showed earlier, and I have circled out New Zealand. And you probably have noticed a big difference between these two maps, and the point that I'm trying to highlight, which is that New Zealand does not have any native rhododendron species. But as I have mentioned, New Zealand is the country and the area that has been conserving a lot of Maddenia species, as well as other rhododendron plants. So here, I will briefly introduce the National Conservation Project here and the herbarium documentation and taxonomic identification work that I'm involved in, and also the community networking and education activities happening here. So my guess is that many of you have heard or have traveled in New Zealand, that New Zealand has a beautiful, breathtaking landscape. So people are happy here, and the nature is, so far as I know, is quite happy as well. And I guess the plants are also very happy. As you can see, New Zealand, the whole country lands in the temperate climate zone. So it is very favorable for subsection Maddenia. And the local community here has formed a national situ conservation project for rhododendron with a few botanic gardens from north to south engaged. So here, Pukeiti Gardens, as I'm showing on the right, is the leading garden who also sponsored my PhD research. And one highlight that I'd like to show you is that at the back of this garden, as you can see, there are some mountains, and it is actually a volcano. So this garden sits in the corner here, and the dark dot area on the map is actually this volcano. So there are some fertile soils here, which is very helpful for growing rhododendrons, and this is an alpine area. And the rhododendron plants are quite happy here, and Pukeiti has contributed the majority of the subsection Maddenia collections in the conservation project. And the local community here has been propagating and reproducing many of the threatened Maddenia species and distributing them across the whole country. Quoting the leader of this project, who is also the manager of Pukeiti Gardens, Andrew Brook, people have a beautiful dream and very genuine goal for this conservation project. They said, we hope one day to successfully return some of our plants to their countries of origin. So this is exactly what we want for the integrated conservation for plant species. We would like to back up the plant collections for these species in our ex situ collections, so that we will have sufficient material in case one day we need to return them back to the countries of origin or plant them back into the natural habitat. And while we are working on the living plants, we have been also collecting specimens and then identifying the plants that are growing in our gardens. So this is the herbarium documentation work that mainly done by myself, and I also pressed these voucher specimens with a lot of help from the local gardens here and the community who have helped me to collect the specimens. So one of the highlights when I was doing the herbarium documentation is that the Maddenia flowers, they are very fragrant. Even if you have transferred them from different newspapers again and again, you can still smell that fragrance in the herbarium. And even if they are dry like this, for the fragrant plants, you can still get that fragrance out of the dried material. So this was actually a very mentally rewarding work. So with the taxonomic identification work, and also with the assistance from my phylogenetic analysis, we have actually found a few of the mislabeled living collections. For example, these two accessions that we collected from KET, and I also used them in my phylogenetic analysis. With the combination of morphological identification and the genetic analysis, we figured out that they were actually labeled not correctly. So this is an example of how I have been using the theoretical analysis to suggest what to do in the gardens. So I have suggested potatic gardens. Okay, we have these accessions labeled wrong, and I would highly recommend you to make a correction on the labels for these plants so that we actually protect the correct species in our collections. And this whole project and the conservation work is not only done by people who are studying like me, but with the very wide community. The main association involved here is the New Zealand Rhododendron Association, with a lot of enthusiastic members. The picture on the left is from one of the field trips that we visited, and as you can see, how the visitors and the members of the association are enjoying the glamour of all these rhododendron flowers and how careful the local people have been cultivating these plants. So through these field trips, we have been exchanging ideas on how to cultivate rhododendron plants and how to make our contribution to the rhododendron conservation here in New Zealand. And in the picture on the right, that is a presentation that I was invited to give to the New Zealand Rhododendron Association based on the research that I did for my PhD and also some of the recommendations that formed from my research. So I believe with the contribution from the whole community, I think many more of the rhododendron species will be better safeguarded in our garden, in our botanical gardens, and they will have a bright future for their survival, and we will have more material backed up in our botanical gardens and then be secured to return them back to the origins, the countries of origin. So that's all that I wanted to share with you. And here is a list of the references from my whole talk. And if you would like to dive into more details of the information. And lastly is a few pictures that on how I worked with these lovely and very enthusiastic people who are many from the Garden Trust, from the Rhododendron Association, from my supervisor from my supervisor panel, and also some passionate local gardeners. And I do believe that we will have a bright future with a lot more contribution from all of you. And feel free to get in touch if you would like to contribute in any information. Thank you. And
Thank you, Ling. I feel inspired. And you already have a question. Let's look at the Q&A.
yes, please.
So I'll read it. Tara Spears asked, does Phuket Gardens have a policy or criteria that provides guidance as to what they introduce into the gardens? How many, etc.?
Pardon, could you repeat the question?
Yeah. Does Phuket Gardens have a policy or criteria that provides guidance as to what they introduce to the gardens? For example, how many? How many?
Yeah, so I don't have a particular number on how many of all the rhododendron species that can serve, but simply for the Maddenia group, they have 23 out of the 65 taxa conserved here, which is like the sixth largest number worldwide. Yeah, and also the Dunedin Botanic Garden contributes 27, so together they would have the majority of the Maddenia taxa covered here. And in terms of the policy, Phuket, as far as I know, they have signed some agreements, for example, with the Kunming Botanic Garden, and to introduce some more wild material to back up them here, although it's a long and tedious process. But there will be also like the biosecurity department involved here, because New Zealand's natural environment is very fragile and it has a super restrict biosecurity standards. So there's a lot of caution to grow exotic plants here. And also, my supervisor, Mary Mackay, has been working on a whole list of the exotic plants growing in New Zealand, including the rhododendron species. So, for example, from my study, I got to know there was a couple of species that are actually not listed in the Department of the Conservation plant list as considered, okay, they're actually living here in New Zealand. So our work is to secure the place for these plants and let people know they're actually threatened and they are not causing trouble to the natural habitat here in New Zealand. So there's a lot of specific criteria, and we would need to have a much longer conversation on how people are actually introducing these plants and how to have their standard on propagating them and then distributing them following a particular principle. Yeah.
Thanks, Ling. Yeah, so just I'm kind of looking at that garden as a possible structure for our at Jenkins. So, some guidance for our how we like introduce new plants or introduce specimens to our germplasm project. Kind of so. I mean, right now we kind of just have some like a loose guidelines that we came up with ourselves, but I would love to be pointed in the direction of like a garden that is doing this, and they have like their policy and kind of their structure and criteria to, what gets introduced and how many, etc. So yeah, so I appreciate your talk on this. And if you have any, and yeah, I agree. I think that we can definitely have a lot to talk about outside of this. So thank you.
Yeah, thanks, Tara, for your question. And I think a quite different thing is in the US you have many native species, and you could distribute the plant material from one region to Jenkins Botanic Garden, and that doesn't have that super many biosecurity issues, while as here in New Zealand, yes, so we have
Yeah.
to introduce from another land actually.
Uh-huh.
Yeah,
Yeah.
I think yeah.
Oh, go ahead.
Yes, I think we could also get in touch with Andrew Brook. Andrew Brook, yes. So he was there in the ARS conference last year as well from New Zealand. So we could definitely talk about this more in another conversation. Yeah,
Okay, perfect. Thanks, Ling, and nice
yeah.
work.
Thanks.
All right, well, I guess that's what this is for. So you guys can find out that you want to talk more later, right?
Yeah, networking, share knowledge, and exchange ideas.
Yeah. Ling, that was a really fun talk. I really appreciated it. And I'm looking forward to actually watching it again because I think there's so many questions that I had. So
I know.
I would love to be able to figure out how our work can contribute to what you guys are doing. So we don't do genetics or field work, but I think we have a place in the world of conservation. So we're going to find it.
Yes, definitely. Absolutely. Yes. And yeah, please get in touch. And I know there's a whole lot of information in this talk, so
Yeah.
I've hidden a lot of details so that you don't get bored. But yeah, if you are interested, go to the references, and I have 3 of my 4 publications open to the public already. And yeah, we can chat about it later.
Great.
Yeah.
So the talks are going to be up starting on June 1st on YouTube. And so start looking for them on YouTube on June 1st so you can see what you missed and watch the ones you liked again. So thanks, Ling. I think we're going to go on to the next section.
Thanks, Juliana.